So it is my great pleasure to welcome you to this meeting of the GEOTOP-A seminar, and welcome particularly Iris Yoon from Wesleyan University, who's going to give us a lecture on topological tracing of encoded circular coordinates between neural populations. I look forward very much to hearing your talk, Iris.
Thank you, Catherine. So thank you to the organizers for inviting me, and thank you all for being here. As Catherine mentioned, I'll talk a little bit about topological methods that allows us to compare circular coordinates that you might see in different neural populations. So before we do that, let's talk a little bit about why you would even expect to see circular coordinates in neural populations. So here's a cartoon example. Let's say I've measured spike trains, so activities of my neurons throughout some kind of experiment. So here, the x-axis, you should really think of them as time. And wherever you see a vertical line, it's an indication that this particular neuron has fired around that time. So if you see some time intervals with a lot of these vertical bars, it means that that neuron was very active during that time. And if you see more sparser regions, it's indicating that your neuron did not fire heavily. So let's say I've measured these neural activity throughout my experiment, and I wanted to arrange these neurons based on how similar their spike trains are. If you stare at them a little bit long enough, you might say, okay, well, each pair of neurons that I see on the left-hand side are similar to one another, whereas maybe this dark red neuron is more dissimilar to the green neurons on like the fifth row. So if you were to arrange these spike trains according to their similarity, you might end up arranging them in a circular fashion, as shown here. That also means that once you compute the similarities between your neurons and compute your similarity matrix and apply persistent homology, the output of persistent homology should indicate that there is some kind of circular structure among these neurons. And let me give you some other examples, like actual neural systems where we expect to see such circular coordinates. So one example is in the visual system. So let's say I presented these different orientations of images to a person or to a rat. The fact that you have different orientations tells you that your stimulus itself can be arranged in a circular fashion. So the collections of different orientations form one circle. Let's say that as you show these visual cues, you also measure neural activity from neurons that respond to very specific orientations. So maybe the right-hand side shows you some cartoons neural activity in response to such visual cue. If you were to arrange the spike trains according to their similarity as well, you might end up arranging them in a circular fashion. So the idea here is that the visual system, the visual stimuli, the different orientations themselves are arranged in a circular fashion. And because we're looking at neurons that are responding to the different orientations, if you look at their spike train activity, they are also going to encode some kind of circular structure. And this is really being driven by the fact that the visual image, the video themselves, have a circular feature. So the neurons are really picking up the circular feature in your stimulus. Another example is the head directions. So imagine that your mouse has some fixed point, and then you have these neurons that fire according to the angle that your head would make with respect to this one fixed point. The different head directions are then going to, the different possible head directions then range from 0 to 2 pi. And so the collections of head direction cells would also be arranged in a circular fashion. Let's think about other neural systems. So now we might not necessarily see like circles that are intrinsic to the system, but may be driven by an environment. So here, as part of the navigational system, we have these cells called place cells that fire in response to the subject being at a very specific location and place. So here, this cartoon illustrates that we have this one place cell shown in orange that fires as this mouse comes in proximity to this little orange area here in the location. And here's an example of how these place cells fires. So the different colors here indicate different place cells, so different neurons that fire in response to very specific locations in space. And as this mouse is moving around, the colored locations indicate the spots that causes the neurons to fire. So you can see that this bottom right corner causes the red neurons, the red place cells to fire heavily, whereas the center of the room causes the yellow neurons to fire heavily. Oh, and I may have duplicated this video. And these have been like, so the place cells, the neurons that fire in response to very specific locations in space can then be used to recover the homotopy type of the environment that your mouse is moving around. So consider this very simple experiment where you have two environments, one with a hole in the middle and one without. And the idea is that you let your mouse move around and you measure the activity of place cells as this mouse moves around in space. The actual measurements that we're going to get are going to be of these spike trains. And the idea is that you can use the similarities between these spike trains, as I've illustrated before, to construct a simplicial complex whose homotopy type can recover the homotopy type of the two environments shown above. So the idea here is that you use the spike trains. Each spike train or neuron can be considered as a vertex in your simplicial complex. And if you have a collections of neurons that are very similar in their spike train patterns, you add simplices to indicate that these are collections with high similarities. And as you can see from this cartoon drawing on the right-hand side, the simplicial complexes that you build out of the spike trains shown in row B are going to be good approximations of the spaces you can construct out of the receptive fields of your neurons. And it's actually the nerve lemma that we all know and love that allows us to say that the simplicial complexes that you build out of the spike train patterns can recover the homotopy type of the environment that the mouse is in. And really, the idea here is that if you have your stimulus space or the environment that you're moving around, if your receptive fields form a good cover, then the simplicial complex you build out of the receptive fields are going to be homotopy equivalent to the environment that you started with. And the idea here is that if you measure the spike trains of your neurons and measure their similarities and then build your simplicial complex, this should be a good approximation to the nerve or the top simplicial complex that you built out of the good cover. And so really the idea here is that using the spike trains and maybe a good notion of what it means for two spike trains to be similar, you can then reconstruct the homotopy type of your environment. Let me introduce one more types of cells because these are very cool. And these are called the Ghrist cells. So they're also part of the navigational system like before, except that instead of a single neuron firing in response to a very specific one particular location in space, the Ghrist cells now fire in response to multiple locations that are spread out along the space. And the locations that causes these Ghrist cells to fire are organized in a very rigid way. In fact, the locations that causes a single neuron to fire are organized in this kind of hexagonal patterns. So these specific navigational system neurons are called grid cells. And here are some examples of the receptive field or the specific locations in the two-dimensional environment that causes these grid cells to fire. So you should really think of one box as representing one grid cell. The yellow highlights are the locations that causes this grid cell to fire. And when we measure the grid cell activity, we will again see some kind of spike chain as shown here. So why is topology involved with these grid cells? To illustrate the idea, let me just go down a dimension for now, just to maybe 1 dimension. And let's think about what a one-dimensional grid cell would do. So let's assume that my mouse now lives in a one-dimensional line and is only allowed to move forward and backwards. The grid cells in this case are going to be neurons that fire in response to specific locations on the line that are at regular intervals. So the black dots indicate the locations that would cause one of the neurons to fire. Maybe you have another grid cell that's indicated by the red, yellow, green, and so on and so forth. Now let's imagine how these grid cells would fire as the mouse moves on this one-dimensional line. The mouse can move back and forth along the line, but just for simplicity, let's assume that the mouse moves from left to right. So as it moves from left to right, your black grid cells might have this kind of firing pattern, indicating that it has high firing when the mouse is above the specific black dots that's shown here. And similarly for red, yellow, green, and other grid cells, you'll measure your spike trains. If you then try to, again, arrange these spike trains according to their similarities, you might end up arranging them on a circle. And so if you were to compute the similarity matrix and apply your persistent homology algorithm, the output will then again tell you that there is some kind of circular feature among the spike trains. Let's now go back to the two-dimensional grid cells that we're interested in. So here we have, let's say this represents one grid cell's firing location that are all arranged in a hexagonal pattern. Here's another example of a grid cell, and so on and so forth. Then the idea is that if you think about the independent, if you think about, let's say, just looking at one independent direction, so like one-dimensional line, you can see that there is a repeat of grid cells that would fire as you move along this one-dimensional line, right? You see the black, red, orange, and this pattern is going to repeat. So we're really in the situation of looking at one-dimensional grid cells along one line that we've seen a minute ago. And there are two independent directions that would incorporate these one-dimensional circular structures. So if you think about the grid cells, there are two independent directions that have this kind of periodicity. And so the grid cells themselves are then actually arranged on a torus with two independent circles. And indeed, it has been experimentally verified that if you measure the spike train's activities from the grid cells and apply persistent homology, that the output that you get is in fact consistent with that of a torus. So those were some of my favorite examples in the brain or the neural systems that have topological features. So let's get to the main topic of the talk, which is how do I compare neural encodings that might occur in multiple different systems? So the motivation here comes from, again, some neuroscience experiments. So let me illustrate what these experiments look like in cartoons. So consider this hypothetical experiment where you present some kind of visual stimuli and measure activity from some population of neurons. Just from looking at the stimulus alone, you might be able to apply your persistent homology pipeline, which then tells you that there are, let's say, 2 circular features. You can also apply your persistent homology to the measured spike trains, which would then also indicate the circular features that are present along your neural activity. Now the question is, how are the circular features in the stimulus space related to the circular features in the spike trains? In this particular example, is the single circular feature among my neural activity picking up one of the circles that are present in the stimulus, or are they independent? Or is the neural activity actually representing some combination of the circles that are present among my stimulus? Here's another example where this question of comparing neural activity across different neural systems come into play. Here, let's assume that we measure neural activity from 2 different regions of the brain, region A and region B. You can again apply persistent homology on the spike trains in region A alone and region B alone, which will individually indicate some presence of circular features. How do I know whether the circular feature encoded in region A is related to the circular features encoded in region B? Is the circular feature in region B merely a reflection of one of the circular features in region A, or are they, again, completely independent? So in both of these hypothetical experiments, we're trying to compare 2 different barcodes. In particular, we're trying to understand how a circular feature in one system is manifested in another system. And under the hood, really what we're saying is that we have 2 different point clouds, P and Q. In this particular setting, the points represent neurons. And we want to compare the resulting barcodes. Now, if your underlying systems or your underlying point clouds can be embedded into some common space, or if there is a mapping between them, then there are other methods, like other topological and geometric methods that can be used to address this problem. But in these neuroscience experiments, especially the first experiment where we were comparing stimulus spaces and neural activity on one hand, it's really difficult to find such a common embedding that respects the metric in both systems. So our approach to this problem is to, instead of trying to find a common embedding, we're going to use some notion of similarity between the 2 systems. And I will explain how we do this for inherently very distinct systems like stimulus spaces and neural activity. And the key construction that we use here are called witness complexes or Dowker complexes. So let me give a very brief reminder of what these complexes are. So let's say I have 2 different point clouds, one called the landmark and another called witnesses. And let's say I have a matrix measuring similarities or dissimilarities between my landmarks and the witnesses. A witness complex is a simplicial complex that has the landmarks as the potential vertex set. And if I were to build a witness complex at a particular parameter, let's say epsilon, what I do is I add simplices, so simplices consisting of only the landmark points. If the corresponding landmark points have a common witness, i.e., if there is a point among the witnesses that is close enough, where close enough here is governed by the parameter epsilon to all the landmark points that are involved. So here are some examples of the simplices that you might see in your witness complex. And here's an example for this cartoon example of a witness complex at a very specific epsilon parameter. Since there is this parameter dependence, in order to avoid choosing an arbitrary epsilon, what you can do is to construct these witness complexes for various epsilon parameters, i.e., as you vary the similarity requirements between the landmarks and the witnesses. And these witness complexes that you construct are all going to be nested. So in particular, you can compute your homology here as well. The barcode that we get from the filtration of the witness complex will refer to this as a witness barcode or sometimes a Dowker barcode. So let's say we have our two systems or our 2 point clouds, P and Q. You now have to make a choice of whether you want to use the P's as your landmarks or your Q's as your landmarks. And depending on which one you choose, your witness complexes are built on 2 different point clouds. And Dowker's theorem tells us that the choice actually does not matter. So for each column on the screen, the simplicial complex you see on the top and bottom are going to be homotopy equivalent. And the functorial Dowker's theorem says, actually, even if you consider this in the persistence setting, the choice doesn't matter. So all the diagrams that you see here are going to commute up to homotopy. And in particular, if you compute the barcode, the witness barcode from the top and the bottom filtration, they're going to be identical. And the way we think about the witness barcode here is we think of it as picking up some kind of common or shared feature between your two systems. So here's an example of the very explicit cycle representatives that correspond to the one long bar in the witness barcode. And you can see that they represent this pink cycle that is shared between my P's and Q's in this top left corner of my point cloud. So how do we actually use Dowker, this witness barcode and Dowker duality? The method that we propose, we call it the method of analogous bars. And our input is given as following. We have 2 point clouds, P and Q. They don't have to be embedded in the same space. We have the internal distance matrix between among the P's, among the Q's. So just the information that we would normally expect before we can apply persistent homology in the metric space. We are also going to assume that we have some measure of similarity between the points P's and Q's, and these are shown in the pink rectangular matrices. Just as a running example, let's keep this example in mind where my points Q's are sampled from a square torus and P's are sampled along one of the essential circles on my square torus. And again, P's and Q's don't have to be embedded in the same space, but we'll keep this example for illustrative purposes. I can use the distance matrices among P's, distance matrices among Q's to compute my usual barcode from a Viator filtration. And here, all the barcodes are shown in dimension 1. And as you would expect, the barcode among the orange points is going to have one circular feature. The barcode among the Q points are going to indicate that Q has 2 circular features on the square torus. And the question is, I want to understand how these 2 barcodes are related. In particular, I want to understand how the long bar in the top barcode is manifested in the bottom barcode. I want to understand how the single circular feature among the P's is manifested among the Q's. Like which of the 2 circles on my square torus is represented by the circle among the P's. So because we have this cross-system dissimilarity matrix between P and Q, we could compute the witness barcode that I've introduced before. And the long bar in the witness barcode indicates that there is some kind of common feature between the P's and Q's. And the method really relies on making pairwise comparisons between the barcodes that you see on the screen to understand how the top and the bottom barcode are related. So to compare the two witness barcodes that we see in the center, we use Dowker's theorem, which just tells us that they're going to be identical. And then to compare the witness barcodes in pink to the Viator-Scripps barcodes on the top and bottom, we use a method called persistent extension, which we developed for this purpose. So I'll come back to this slide at the end, but for now, let me focus on what persistent extension does, because this is where a lot of the technicality lives. So the goal of persistent extension is really, given a bar in, let's say, the witness barcode highlighted here in pink, I want to understand how this is represented in the Viator-Scripps barcode downstairs. And the witness complexes and the Viator's complexes here, they're actually built on a common vertex set. So here, they're both built on the points Q's, and they're just two distinct filtrations. Now, if you are, the usual way for comparing witness filtrations and Viator-Scripps filtrations is using this relation that I've shown here in the slide. But if you try to use the fact that a witness complex at a particular parameter is contained in the Viator-Scripps complex with the doubled parameter to try to understand how you might relate the bars in the two barcodes, what ends up happening often is that the moment you double the parameter for the witness complex, you end up trivializing a lot of the cycles that you're interested in. So here, because my points P's and Q's are indeed embedded in the same space. In this particular example, this doesn't occur, but when we try to use this approach with neuroscience experiments, we ended up, we realized that the moment you double your parameter of the witness complex, you end up trivializing a lot of the features and you can't really say much about how the bars are related. So to illustrate how we do this comparison between the witness barcode and the Viator-Scripps barcode, just remember that these are two different filtrations built on the same vertex set. And we have these two barcodes. We have a bar, one long bar on the top filtration, and we want to understand how this is manifested in the bottom barcode. And we break up this process into three different steps. In the first step, we find a specific homology class that represents the highlighted bar of interest. In the second step, we find homology classes that live in simplicial complexes in the bottom filtration. And then on the third step, we find its representations in the barcode downstairs. So I'll briefly go over how each of these steps work. So in the first step, we're given a bar of interest and we want to find a corresponding homology class. So to do this, what we do is instead of looking at the entire filtration, we fix a parameter. And this parameter is chosen to be the parameter immediately prior to the death time of the highlighted bar. And we fix the simplicial complex at that point. And then we find a homology class that represents this bar at that specific simplicial complex. And we choose the parameter to be immediately prior to the death time of the bar, because this is the point where my simplicial complex has as many simplices as possible without trivializing the cycle. So this is the point where my simplicial complex has as many cycle representatives representing the same bar. And if you're computing your homology with field 2 coefficients, you can show that the homology class that you identify this way is unique. And it doesn't depend on the choice of bases of the persistence module that you started with. So in the second step, we look for homology classes in the bottom simplicial complex. Now at this point, remember that we started with two different filtrations, and we managed to fix a simplicial complex for the top filtration in a unique way. And now I have to find homology classes in the bottom filtration that represent the same cycle as the ones shown above. Now, if I had a much simpler setting, so for example, let's assume that instead of having an entire filtration in the bottom, I only had a single simplicial complex to think about, then finding similar homology classes in the bottom is quite easy. One way to do this is you just build some kind of intersected complex. You find if there is a non-trivial homology class in the intersection, so shown here in orange, that maps to the pink one on top. You can also look for the image of that orange class to the bottom. Then that image shown here in navy is going to be very similar to the homology class on the top. So you can use this method to look for homology classes in the bottom complex that are similar to the one on the top. And this is not unique. There might be multiple of these, and that's okay. Our method, because we don't want to make any arbitrary choices about which homology class is most similar to the one on the top, we actually end up returning all of these as valid similar homology classes. And finding these really, it turns out to, you just have to solve some kind of linear system of equations. Okay, so in the simpler setting where I have a fixed simplicial complex in the bottom, finding related homology classes in the bottom complex that's similar to the pink cycle above is quite simple. You just have to solve some linear system of equations. But remember that we actually have an entire filtration to consider. And the naive way to go about this would then be, okay, for every single parameter on the bottom filtration, I can try to build this intersected complex and then solve my linear system of equations at every single parameter. But that would be infeasible because I would have too many parameters to check. So the way we addressed this challenge was we noticed that when you build your intersected complex, these are also themselves going to be nested. And so in particular, if you find the solutions to these particular linear system of equations at a particular parameter, those are going to continue to be valid solutions at later parameters as well. So what that tells us is that you could try to identify very strategic parameters at which you need to solve that particular linear system of equations and then recover all the solutions at all the other parameters. So I won't go through like how we exactly do this here, but the idea is that instead of looking at every single parameter, you can parse this intersected complex filtration to identify strategic places, strategic parameters at which you need to solve this linear system of equations. And then you can recover all the solutions at all the other parameters. And this gives you a huge computational advantage because now you've dramatically decreased your search space. Or sorry, the parameters that you need to solve the system. So during the second step, we implement this method. Again, the idea is that you look at this new filtration you get from looking at the intersections. You solve some linear system of equations, and that gives you a collections of homology classes in the bottom filtration that are similar to the pink cycle that we started with. And then the last step is for each homology class that you just found on the second step, you represent it using the barcodes downstairs. And for this, if you have a fixed bases for your persistence module. This is quite easy. You just have to find the bars that represent the homology class that you found using that very specific basis. But as we all know, the question of finding an optimal basis for a persistence module is quite challenging. And so in our method, instead of fixing a particular basis that we think is optimal, we actually allow the user to experiment with different types of bases and see how the homology class that you started with is manifested as a collection of bars in your barcode. So to sum up the persistent extension part of the work, we had a bar of interest in one barcode. In our case, this came from the witness barcode. We find a corresponding homology class that represents this bar. We find a collection of homology classes that are similar to the pink class identified above. And then we represent those navy classes as collections of bars in our barcode. And again, really, this process was motivated by this problem of how do you compare two different filtrations built on a common vertex set. So here's, let's go back to the running example that we had. So remember, Q was sampled from a square torus, P was sampled along one of the essential circles. The pink barcode here is the witness barcode, and the green barcode on the bottom is the Viator strips barcode, and they're both built on Q as the vertex set. So we have this one highlighted bar in the witness barcode indicating that Q and P have some kind of shared feature. If we use the persistent extension method, the homology class that represents this highlighted bar is going to be this horizontal S1. When you find the corresponding homology classes in the Viator strips complex, they are also going to be S1, the horizontal S1s as well. And these are going to be their barcode representations. Now, when you look at the highlighted bars in the bottom, you'll notice that both of these highlight one of the long bars, and it's going to also highlight some of these like shorter bars as well. So what we've observed is that for the different choices of the similar homology classes, so here we show two different choices, there are always going to be some combination of this one single long bar, this one particular long bar, and some of these other shorter bars. And indeed, if you go through the cycle representatives for this very long highlighted bar, this is going to be the bar that represents the horizontal S1 in your Viator strips complexes. So coming back to the original problem where we had two different systems, P's and Q's, and we wanted to compare the barcodes in the very top and the bottom, what we do here is we're going to make pairwise comparisons among the barcodes that we see on the screen. So one way of doing this is you start with, let's say, one highlighted bar in one bar of interest in your top barcode. So here we're highlighting the one circular feature among the P's. We use this persistent extension method to find how that bar is manifested in the witness barcode. We then use Dowker's theorem to find this representation in the dual complex and the dual witness complex. And then we use persistent extension again to find this representation in the very bottom barcode. Another way to go about this problem is we start with the witness barcode in the center. So the fact that we have one long bar in the witness barcode indicates that we have some kind of shared feature among P's and Q's. We start with that, and then we use persistent extension to find this representation and the very top and the bottom barcodes. And the bars that have been identified at the top and bottom this way, we refer to those as analogous bars. And indeed, if you actually do look at their cycle representatives, you can verify that the bars that have been identified are all indeed tracing this kind of horizontal S1 on your square torus. So I've started this talk with some neuroscience motivations. So let's discuss how the method is applied in those settings. So recall that I introduced this talk with this problem of comparing barcodes you get from the stimulus space and also the barcodes you get from the spike trains. Another problem of interest was when you have barcodes obtained from two different neural populations, how do you compare them? So we've done a series of simulation studies and we've implemented our method on experimental data sets as well. I'll briefly talk about how the method performed in the simulation study, because that's where we could actually do really rigorous proof of concept and check that the method performs as expected. So we first created this visual stimulus consisting of a circular mask with a orientation, and every few frames, the circle moves around, like it changes the movement direction, and it also changes its orientation. And you notice that the top and bottom edges of the image and the left and right image of the left and right edges of the image are identified. So my stimulus actually lives on a two torus. If you think about the collection of images that are involved in the stimulus and consider the space that they live in, they're going to live on a three torus. One of the circles comes from the fact that my orientation is on an S1, and then the other two comes from the fact that my image itself is living on a torus. We then simulated neurons that would respond in response to this stimulus using Gabor filters. And we simulated their spike trains. Because we designed these neurons to respond to the stimulus that we designed earlier, the neurons are also going to live on a three torus, because we're going to have collections of neurons that pick up the orientation information, and we're going to have collections of neurons that pick up the fact that our image lives on a square torus. So indeed, if you apply persistent homology to the collections of images, you're going to get a persistence diagram that tells you that there are three circular features. And similarly, if you apply your persistent homology to the collections of spike trains from the simulation, the output is going to tell you that there are three circular features. And all of this is again computed in H1. Now, if you apply the method of analogous bars to this data, it's going to, the output is going to return pairs of points in the stimulus persistence diagram and the neural activity persistence diagrams according to these colors. So the points on the left and right diagrams with the same color are returned as analogous points or matched points under our method. So it's going to return that there are three pairs, the pink, yellow, and purple, that are analogous to each other in the two different systems. And indeed, when you go back and investigate the cycle representatives of each of these points, you can verify that the pink points in both the stimulus and the neural activities do indeed both correspond to this orientation's cyclicity, and that the yellow and the purple points correspond to cyclicity coming from the square torus. And we could, in fact, investigate the cycle representatives in this case because the neurons that we simulated were simulated according to these Gabor filters. In an experimental setting, when you do look at the cycle representatives, you're going to have a collections of very long spike trains, and you wouldn't be able to make sense out of it. Okay, we then took the simulation study a little bit further, and we simulated some downstream neurons. So we simulated a collections of neurons that are sensitive just to the orientations, and these were designed so that they did not have access to the original stimulus video. These orientation neurons were trained using a neural network that took the V1 neuron spike trains as input. So they only had access to spike trains from the original simulation and not the video themselves. Because these neurons were designed to pick up the orientation information, we expect them to live on a S1. We also designed another collection of neurons called the direction neurons that are sensitive to the movement direction of my stimulus. Now, again, the direction neurons didn't have access to the original video. They were trained using a neural network that took as input the spike trains of my V1 neurons. And in particular, the neural network would look at time intervals, like some time intervals of the V1 neuron spike train to deduce the movement direction of my stimulus, even though it did not have any access to the original video. So if you think about the space that the direction neurons live in, they're also going to live on S1 because the different directions that you can move into also form a circle. So as expected, if you run persistent homology on each of these collections, you're going to get a persistence diagram that tells you that there are single significant features. If you apply the analogous bars method to compare the persistence diagrams of the V1 neurons to the orientation neurons, it's going to identify this single point in the orientation persistence diagram as being analogous to the pink point in the V1 neuron persistence diagram. And again, if you go through the cycle representatives, you can verify that the pink points are indeed the points that encode orientation cyclicity in my stimulus. For the direction neurons, note that the movement direction is, you can only deduce the movement direction if you consider some kind of time interval. So this is like dynamic information that is not, that's not present if your neurons are only designed to respond to static information like orientations and locations. So the V1 neurons were designed to be to pick up very like static information. And so the V1 neurons have no idea, they're not able to encode any kind of like movement direction information. Whereas the direction neurons were designed to look at time intervals to pick up the movement directions. So the idea is that the circular information encoded in the direction neurons, they're not present among the V1 neurons shown here. So indeed, when we apply the analogous bars method, our method tells us that the single feature that you see among the direction neurons is independent of the circular features that are present among the V1 neurons. And that is actually quite critical in that our method isn't just trying to align the geometry as much as possible, or it's not trying to match the points on the persistence diagrams as much as possible, because they would result in this teal point being matched to one of the points on the V1 neuron persistence diagram. Our method concludes that these two are actually independent circular features, and that is exactly how we want the method to perform. So in summary, I presented the method of analogous bars that allows us to compare barcodes from two systems when the two systems can't be embedded into a common space. And the key idea here was really using witness complexes or Dowker complexes to find some kind of shared or common feature. And I did not go through all the simulations and the experimental study that we've done, but we've done some proof of concept showing, really verifying that the method performs as we expect, i.e., if there are circular features that are related, that our method picks them up, and that if you do have independent features, then our method will not identify them as being related. So that is all. Thank you for your attention, and please let me know if you have any questions.
Well, thank you very much, Iris. Does anyone have a question from the audience? Well, I have a question, but it's a very curiosity. Is how can we interpret this relationship between the cycles? Is it so the fact that you have a cycle in one set of neurons and another cycle in another set of neurons, how can we interpret that? Is it that some neurons can replace the other ones, the function of the other ones, or probably something completely different? I don't know.
Good question. So it means that your two different systems are encoding some shared information. So it could be that in this simulated system that we've designed, what is indicating that is that there are three circular information that's encoded by one population of neurons, and one of them is being projected down to another population of neurons. Whereas you don't see any relations, as you see here between the V1 neurons and the direction neurons, then that's an indication that the information encoded by the direction neurons is probably a result of some kind of higher level computations, or they're just picking up something completely different from what's being encoded by V1 neurons.
Okay, and somehow when they are related, that will means that they are like extracting some kind of or how can you say summarizing the information that the other neurons computed or something like that?
Yeah, so they're encoding the same information. So here in this particular simulation, the orientation neurons got information from the V1 neurons. So you can think of this as, okay, there are three information that's encoded by the V1 neurons. Only one of them is being sent to the orientation neurons. But it's also possible that your two neural systems are very independent, like there's no information flow between those two. So in that, it would imply that your two neural systems they're encoding the same information, even if there is no information flow between them.
Oh, I see. Oh, thank you very much. Are there any other questions from the audience? Well, if there is not, well, I'd just like to thank you again, Iris, for your amazing talk. And it's a pleasure to have you here.